Hello there, and welcome back to Bedtime Science. I'm really glad you're here tonight. The body you are lying in right now is not a simple object. It is a living system made of roughly 37 trillion human cells, surrounded by trillions of microbes, powered by chemical reactions, governed by electrical signals, repaired in the dark, defended without your awareness, and coordinated with a precision that no single conscious mind could ever manage. Your heart is beating. Your lungs are exchanging gases with the air. Your kidneys are filtering your blood. Your liver is transforming chemistry on a scale that would overwhelm any laboratory. Your immune system is inspecting the borders. Your nervous system is turning sound, touch, memory, and thought into the private experience of being alive. The human body is one of the most familiar things in existence because every person has one. And at the same time, it is one of the strangest. The closer science looks, the less mechanical it seems. It is not a machine in the simple sense. It is an organized storm of cells, signals, fluids, hormones, microbes, tissues, instincts, rhythms, repairs, and ancient evolutionary compromises. It is full of systems that began hundreds of millions of years before humans existed, refined by trial, survival, failure, and adaptation across deep time. Tonight, we move slowly through that hidden world. The cells, the blood, the heart, the brain, the senses, the immune system, the gut, the skin, the bones, the chemistry of sleep, and the quiet work the body continues to do while the conscious mind lets go for the night. The human body contains approximately 37 trillion cells, a number estimated through careful measurement of cell sizes and tissue densities across different body compartments. These cells are not all the same. The body contains approximately 200 distinct cell types, each with a specific structure, function, and lifespan. Red blood cells, which carry oxygen through the bloodstream, have no nucleus and live for approximately 120 days before being broken down and recycled. Neurons in the cerebral cortex can persist for the entire human lifetime without being replaced. The cells lining the small intestine are replaced every 3 to 5 days. Liver cells turn over approximately every 12 to 18 months. Bone cells live for approximately 10 years. The body is not a static structure, but a continuous process of renewal, with different compartments operating on different timescales of replacement and regeneration. The human genome, the complete set of genetic instructions encoded in the DNA of each human cell, consists of approximately 3 billion base pairs arranged along 23 pairs of chromosomes. If the DNA from a single human cell were stretched out end to end, it would extend approximately 2 meters in length. The DNA in all of the cells of one human body, stretched end to end, would reach from the Earth to the Sun and back approximately 60 times. This 2 meters of DNA is packed into a cell nucleus approximately 6 micrometers in diameter, about a tenth the width of a human hair, through a complex hierarchical folding process involving proteins called histones around which the DNA is wound, and larger-scale organizational structures that compact it by a factor of approximately 10,000 compared to its extended length. How this compacted DNA is precisely and selectively accessed by the molecular machinery that reads specific genes at specific times is one of the central questions of contemporary cell biology. Only a small fraction of the human genome codes for proteins. Approximately 1.5% to 2% of the genome consists of protein coding sequences, the genes in the most familiar sense. The remaining approximately 98% was once called junk DNA, reflecting an assumption that it was non-functional evolutionary debris. This characterization has been substantially revised by subsequent research. Large portions of the non-coding genome contain regulatory sequences that control when, where, and at what level protein coding genes are expressed. Other regions encode functional RNA molecules that do not produce proteins but play important roles in regulating gene expression, maintaining chromosome integrity, and other cellular functions. The full functional landscape of the non-coding genome is still being mapped, and the proportion that serves no current function remains genuinely uncertain. The human heart beats approximately 100,000 times per day, or about 35 million times per year. Over a typical human lifespan of approximately 80 years, this amounts to roughly 3 billion heartbeats. The heart is a dual pump. The right side pumps deoxygenated blood to the lungs to be oxygenated, and the left side pumps the oxygenated blood from the lungs to the rest of the body, and it generates its own electrical rhythm through a specialized group of cells called the sinoatrial node, located in the right atrium, which fires spontaneously and regularly without any external nervous input. This is why a heart removed from the body continues to beat in the appropriate chemical solution. It carries its own pacemaker. The heart pumps approximately 5 liters of blood per minute at rest, meaning that the entire blood volume of a typical adult, approximately 5 liters, completes a full circuit through the body once per minute. During vigorous exercise, cardiac output can increase to 20 to 25 liters per minute. The circulatory system contains approximately 100,000 kilometers of blood vessels, enough to circle the Earth approximately 2.5 times. This network consists of arteries carrying blood away from the heart, veins carrying blood back, and capillaries, the finest vessels, typically only a single cell wide, where the actual exchange of oxygen, nutrients, and waste products between blood and tissues occurs. The capillaries are so fine and so numerous that no living cell in the body is more than approximately 100 micrometers from the nearest capillary. The total surface area of all the capillaries in the human body is estimated at approximately 6,000 square meters, the size of a large building, providing the enormous surface area needed for efficient exchange across the entire body simultaneously. Blood is a complex living tissue, not simply a transport fluid. A cubic millimeter of blood, a volume smaller than a pinhead, contains approximately 5 million red blood cells, 7,000 white blood cells, and 250,000 platelets, suspended in plasma, the liquid component that makes up approximately 55% of blood volume by composition. The plasma itself contains water, dissolved salts, proteins including albumin and clotting factors, hormones, nutrients, antibodies, and dissolved gases. Red blood cells carry oxygen bound to hemoglobin, a protein that contains iron at its functional core and that undergoes a reversible shape change that increases or decreases its affinity for oxygen depending on the local oxygen concentration, allowing it to load oxygen efficiently in the oxygen-rich environment of the lung capillaries and release it efficiently in the oxygen-poor environment of metabolically active tissues. The lungs are not simply passive bags that inflate and deflate. They are extraordinarily complex structures that maximize the surface area available for gas exchange while minimizing the path length that oxygen must diffuse from air to blood. Each lung contains approximately 300 million alveoli, tiny air sacs approximately 200 micrometers in diameter, each surrounded by a network of capillaries. The total surface area of the alveoli in both lungs is estimated at approximately 70 square meters, approximately the floor area of a small apartment, compressed into 2 organs that fit within the chest cavity. The wall separating the air in the alveolus from the blood in the surrounding capillary is approximately half a micrometer thick, about a 200th the width of a human hair, allowing extremely rapid diffusion of oxygen and carbon dioxide across the barrier. With each breath, approximately half a liter of fresh air reaches the alveoli, and approximately 300 milliliters of oxygen transfer from air to blood per minute at rest. The liver is the most metabolically active organ in the body and performs an extraordinary range of functions that has led it to be described as the body's chemical processing plant. It receives blood from two sources simultaneously: oxygenated blood from the hepatic artery and nutrient-rich blood from the digestive system via the portal vein, and performs over 500 distinct metabolic functions. These include metabolizing and detoxifying drugs, alcohol, and other potentially harmful substances, producing bile for fat digestion, synthesizing most of the proteins in blood plasma, converting excess glucose to glycogen for storage, and releasing it when blood sugar falls, producing cholesterol and lipoproteins, processing and storing fat-soluble vitamins, and degrading red blood cells at the end of their lifespan. The liver has a remarkable regenerative capacity. Up to approximately 70% of the liver can be surgically removed, and the remaining tissue will regenerate to approximately the original size within a few weeks, making partial liver transplantation, in which a living donor provides a portion of their liver, feasible in ways that would be impossible with most other organs. The kidneys filter approximately 180 liters of blood per day, roughly 50 times the total blood volume, through a system of approximately 1 million microscopic filtering units called nephrons in each kidney. The filtration process is selective and precise. Small molecules including water, salts, glucose, and urea pass through the filtration membrane into the tubules, while larger molecules including proteins and blood cells are retained. As the filtrate passes through the tubular network, approximately 99% of the water and all of the glucose are reabsorbed back into the bloodstream through active transport processes. Waste products including urea, creatinine, and excess salts are concentrated in the remaining filtrate, which exits the kidney as urine. The kidneys also produce hormones including erythropoietin, which stimulates red blood cell production, and renin, which regulates blood pressure. The extraordinary precision of kidney filtration, maintaining blood composition within narrow ranges despite wide variation in diet and hydration, is one of the more remarkable feats of physiological regulation in the body. The immune system is not a single organ but a distributed network of cells, tissues, proteins, and chemical signals spread throughout the body. Its primary function is distinguishing self from non-self, recognizing the body's own cells and leaving them alone, while detecting and eliminating foreign organisms, infected cells, and abnormal cells. This distinction is made possible by molecular markers on cell surfaces, human leukocyte antigens, or HLAs, that are essentially a unique molecular identity card for each individual's cells. The immune system has two broad components: the innate immune system, which provides rapid, non-specific responses to a wide range of threats, and the adaptive immune system, which produces highly specific responses tailored to particular pathogens and generates immunological memory, the basis of vaccination and of immunity following infection. The adaptive immune system has an almost unlimited capacity for specificity. Through a process of genetic recombination during lymphocyte development, the body can generate an estimated 10 to the power of 11 distinct antibody specificities, providing coverage for essentially any molecular structure the immune system might encounter. The human brain contains approximately 86 billion neurons, nerve cells, connected by approximately 100 trillion synapses. The brain accounts for approximately 2% of body mass, but consumes approximately 20% of the body's resting energy expenditure, reflecting the enormous metabolic cost of maintaining the electrical and chemical processes that underlie neural activity. The brain does not pause to rest in the way that muscles can. Even during deep sleep, significant brain activity continues, including the consolidation of memories, the regulation of physiological processes, and the generation of dreams. The approximately 100 trillion synaptic connections in the human brain are the physical substrate of everything a person has ever learned, felt, remembered, or thought, a network of such complexity that its complete mapping remains beyond the reach of any current technology. Though the Human Connectome Project and related initiatives are making progress toward mapping the large-scale connectivity structure of the human brain. Neurons communicate through electrical signals transmitted along their axons and chemical signals at synapses. When an electrical impulse reaches the axon terminal of a neuron, it triggers the release of neurotransmitter molecules into the synaptic cleft, the microscopic gap between the axon terminal and the receiving cell. These molecules bind to receptors on the receiving cell, either depolarizing it to make it more likely to fire or hyperpolarizing it to make it less likely to fire. The most common excitatory neurotransmitter in the brain is glutamate. The most common inhibitory neurotransmitter is GABA. Other important neurotransmitters include dopamine, serotonin, norepinephrine, acetylcholine, and histamine, each with specific distributions in the brain and specific functional roles in cognition, mood, and behavior. The drugs that treat depression, anxiety, psychosis, attention disorders, and many other neurological conditions work primarily by modifying the activity of these neurotransmitter systems. The spinal cord is not simply a passive cable carrying signals between the brain and the body. It contains complex circuits that can generate coordinated motor outputs, including the rhythmic movements of walking, independently of descending commands from the brain. These spinal interneuron circuits, called central pattern generators, produce the alternating left-right, flexor-extensor patterns that underlie locomotion, and their activity is modulated rather than generated by descending input from the brain. This organization allows the brain to issue relatively simple high-level commands: walk forward, turn left, while the spinal cord handles the complex timing and coordination of the individual muscle contractions that execute those commands. The reflexes mediated by the spinal cord, the knee jerk, the withdrawal reflex from a painful stimulus, operate at spinal cord level and are complete before information about the stimulus reaches the brain, explaining why withdrawal from a painful stimulus can begin before you consciously feel the pain. Skeletal muscle is one of the most energetically adaptable tissues in the body. At rest, muscles consume relatively little energy. During intense exercise, energy expenditure in active muscles can increase by a factor of 100 or more within seconds, requiring rapid increases in blood flow, oxygen delivery, and substrate availability. This remarkable metabolic flexibility is made possible by the coexistence of multiple energy systems within muscle fibers. The immediate phosphocreatine system provides energy for very short bursts of maximal effort. The glycolytic system produces ATP from glucose without oxygen and supports intense effort for up to a few minutes. And the oxidative system, which uses oxygen to completely metabolize glucose and fat, supports sustained lower intensity activity. Different types of muscle fibers, slow twitch, high oxidative fibers for sustained activity, and fast twitch, high glycolytic fibers for powerful brief contractions, are present in different proportions in different muscles depending on their typical functional demands, and these proportions can be partially modified by training. Bone is a living tissue in a state of continuous remodeling. Despite appearing to be static structure, bone is constantly being resorbed by cells called osteoclasts, which dissolve the mineral matrix of bone and release calcium into the bloodstream, and rebuilt by cells called osteoblasts, which deposit new mineral matrix. This continuous remodeling allows bone to repair microscopic damage before it accumulates into stress fractures, adapt its structure and density in response to the mechanical stresses placed on it, and serve as a reservoir of calcium that can be mobilized when blood calcium levels fall. The balance between osteoclast and osteoblast activity is regulated by hormones including parathyroid hormone, which stimulates bone resorption, and calcitonin, which inhibits it, as well as by mechanical loading. Bones that bear weight or experience mechanical stress maintain their density better than bones in the absence of mechanical loading, which is why astronauts experience significant bone density loss during extended weightlessness and why weight-bearing exercise is recommended for bone health. The skin is the largest organ of the body, with a total surface area of approximately 1.7 to 2 square meters in an adult and a mass of approximately 3.5 to 10 kilograms, depending on how it is defined. It performs multiple critical functions: physical barrier against pathogens, chemicals, and physical trauma, temperature regulation through sweating and changes in blood flow to the skin surface, sensory organ for touch, pressure, pain, and temperature, synthesis of vitamin D when exposed to ultraviolet light, and immune function, with skin-resident immune cells providing a first line of defense against pathogens that penetrate the physical barrier. The skin is a complex multi-layered structure. The outermost epidermis is replaced approximately every 30 to 50 days, with new cells continuously produced in the deepest layer and migrating toward the surface, differentiating and eventually forming the flattened, keratin-rich dead cells that constitute the stratum corneum, the tough outer surface that directly contacts the environment. The dermis beneath the epidermis contains collagen and elastin fibers that give skin its mechanical properties, as well as blood vessels, nerve endings, sweat glands, and hair follicles. The human eye is a remarkable optical instrument operating over an enormous range of light intensities, from starlight to bright sunlight, a range of roughly 1 billion to 1. In dark adapted vision, individual rod photoreceptors can respond to extremely small amounts of light, even to single photons under experimental conditions, though conscious visual perception depends on the signal being amplified and interpreted by the rest of the visual system. The retina contains two types of photoreceptors: rods, numbering approximately 120 million, which are sensitive to low light levels but do not distinguish colors, and cones, numbering approximately 6 million, which are less sensitive but provide color vision. Cones are concentrated in the fovea, the central region of the retina, which provides the highest acuity, color-rich vision used for reading and detailed work. The optical image formed on the retina by the eye's lens is actually inverted, upside down, and left-right reversed, and this inverted image is transmitted to the visual cortex as an inverted neural signal. The brain does not flip the image. Rather, the visual system simply processes the pattern of neural activity from the retina without requiring the image to be physically reoriented. The distinction between the optical image and the perceived image is one of many points at which the experience of vision diverges from a simple camera-like recording. The ear detects sound through a mechanical transduction process of extraordinary sensitivity and precision. Sound waves entering the ear canal cause the eardrum to vibrate. These vibrations are transmitted through three tiny bones, the malleus, incus, and stapes, the smallest bones in the human body, to the fluid-filled cochlea of the inner ear. Inside the cochlea, a structure called the basilar membrane vibrates in response to these fluid pressure waves, with different regions of the membrane responding maximally to different frequencies: high frequencies near the base, low frequencies near the apex, creating a tonotopic map. Hair cells sitting on the basilar membrane detect the mechanical vibrations and convert them to electrical signals that are transmitted to the brain by the auditory nerve. The mechanical amplification of sound provided by the middle ear bones, combined with the sensitivity of the hair cells and the neural processing that follows, allows the human ear to detect extraordinarily faint pressure changes. Around the frequencies where human hearing is most sensitive, The standard reference threshold is about 20 micropascals, a pressure fluctuation so small that it is difficult to imagine in ordinary physical terms. The sense of smell depends on specialized olfactory receptor neurons in the roof of the nasal cavity, which contain approximately 400 types of olfactory receptors, each sensitive to different chemical structures. When odor molecules bind to these receptors, they trigger electrical signals that are transmitted directly to the olfactory bulb in the brain, and from there to the olfactory cortex and the limbic system, the system of brain structures involved in emotion and memory. The direct anatomical connection between the olfactory system and the limbic system is thought to explain the well-known emotional and memory-evoking power of smell. Odors can trigger vivid autobiographical memories and strong emotional responses more reliably than most other sensory modalities. Older estimates often claim that the human nose could distinguish around 10,000 odors, but more recent research suggests human olfactory discrimination may be far richer than that, possibly extending to vastly larger numbers of distinguishable odor mixtures, though the exact number remains debated. The approximately 400 functional olfactory receptor genes in humans represent the largest gene family in the human genome, and this investment reflects the evolutionary importance of chemical sensing. The human gut contains tens of trillions of microorganisms, bacteria, archaea, fungi, and viruses, collectively constituting what is sometimes called the microbiome or microbiota. Older popular accounts often claim that microbes outnumber human cells by 10 to 1 or more, but revised estimates suggest the number of bacterial cells in and on the body is roughly the same order of magnitude as the number of human cells, varying with body size, diet, and recent bowel contents. These microorganisms are not merely passengers. They carry out essential metabolic functions that the human genome alone cannot support, including the fermentation of dietary fibers into short-chain fatty acids that nourish the cells lining the colon, the synthesis of certain vitamins including vitamin K and several B vitamins, the training and regulation of the immune system, the metabolism of drugs and other foreign chemicals, and the modulation of the enteric nervous system, the extensive network of neurons that governs gut function and communicates bidirectionally with the brain through the vagus nerve. The composition of the gut microbiome varies considerably between individuals and is influenced by diet, genetics, early life exposures, and various other factors. Research over the past 2 decades has revealed associations between gut microbiome composition and a remarkable range of health outcomes extending well beyond digestive function. The enteric nervous system, sometimes called the second brain, contains approximately 500 million neurons and is capable of regulating gastrointestinal function entirely independently of the central nervous system. The gut can contract, relax, secrete digestive enzymes, coordinate peristalsis, the wave-like muscular contractions that move food through the digestive tract, and respond to local stimuli without any input from the brain or spinal cord. This autonomy is why gastrointestinal function continues in patients with spinal cord injuries and in isolated gut segments maintained in laboratory conditions. The enteric nervous system communicates with the brain through the vagus nerve, which carries information about the gut's contents and state to the brainstem, and through hormonal signaling. Most of the body's serotonin is produced in the gut, primarily by enterochromaffin cells, where it plays roles in regulating intestinal motility and gastrointestinal secretion. This gut-derived serotonin is not simply the same as brain serotonin, because much of it acts locally or peripherally rather than crossing directly into the brain, but it remains an important example of how molecules associated with the nervous system also have major roles throughout the body. The bidirectional communication between gut and brain, now called the gut-brain axis, is an active and rapidly developing area of research with implications for understanding how gut health relates to mental health and vice versa. The pancreas is a dual-function organ that performs both exocrine and endocrine roles. Its exocrine function involves the production and secretion of digestive enzymes, proteases that break down proteins, lipases that break down fats, and amylases that break down carbohydrates, through ducts into the small intestine. Its endocrine function involves clusters of cells called the islets of Langerhans, scattered throughout the pancreatic tissue, which produce and secrete hormones directly into the bloodstream. The most important of these hormones are insulin, produced by beta cells, which lowers blood glucose by facilitating its uptake by cells throughout the body, and glucagon, produced by alpha cells, which raises blood glucose by stimulating the liver to release glucose from glycogen stores. The precise regulation of blood glucose by this hormonal system maintains blood sugar within a narrow range despite the wide variation in glucose input from meals and glucose consumption during exercise. The failure of insulin production by the beta cells results in type 1 diabetes. The failure of cells to respond appropriately to insulin results in type 2 diabetes, conditions that collectively affect hundreds of millions of people worldwide and illustrate the central importance of this regulatory system. The mechanisms by which the body regulates body temperature are extraordinarily precise and physiologically complex. Core body temperature is maintained within approximately half a degree Celsius of the set point of approximately 37 degrees Celsius, despite wide variation in ambient temperature, physical activity, and metabolic state. When core temperature rises above the set point, the hypothalamus, the brain region that functions as the body's thermostat, activates cooling mechanisms. Blood vessels near the skin's surface dilate, bringing more warm blood to the surface where heat can be lost to the environment. Sweat glands are activated, and the evaporation of sweat carries heat away from the skin surface, and metabolic rate may be modestly reduced. When core temperature falls below the set point, countermeasures are activated. Blood vessels near the skin's surface constrict, reducing heat loss. Brown adipose tissue, a specialized fat that generates heat through a process called thermogenesis, is activated in infants and in cold-adapted adults. And shivering, the rapid, involuntary contraction of skeletal muscles, generates heat as a byproduct of the metabolic inefficiency of repeated contraction and relaxation. Sleep is one of the most physiologically active states the body enters, despite the common perception of sleep as a period of relative inactivity. During sleep, growth hormone is secreted at its highest rates of the day, driving cellular repair and growth. The immune system is particularly active, with certain immune cell populations and immune functions showing peak activity during specific sleep stages. The brain undergoes memory consolidation, the clearance of metabolic waste through the glymphatic system, and a complex reorganization of neural activity. The body's circadian clock, an internal timekeeping system based in the suprachiasmatic nucleus of the hypothalamus, coordinates the timing of hundreds of physiological processes, including hormone secretion, immune function, cell division, and metabolic rate over the 24-hour day. The suprachiasmatic nucleus receives direct input from specialized photoreceptors in the retina and uses light exposure to synchronize the internal clock to the external day-night cycle, a process called entrainment. Disruption of circadian timing through shift work, chronic jet lag, or irregular sleep schedules is associated with increased risk of metabolic disorders, cardiovascular disease, immune dysfunction, and certain cancers, reflecting the fundamental importance of temporal coordination and physiological function. The glymphatic system is a relatively recently discovered waste clearance system of the brain, described in detail for the first time in 2013 by Maiken Nedergaard and colleagues at the University of Rochester. During sleep, particularly deep, slow-wave sleep, Cerebrospinal fluid flows through channels surrounding the brain's blood vessels, driven by rhythmic contractions of arterial walls, flushing out metabolic waste products including amyloid beta protein, the accumulation of which is associated with Alzheimer's disease. This cerebrospinal fluid flow is dramatically more active during sleep than during waking, with the channels through which it flows expanding approximately 60% compared to waking state. The discovery of the glymphatic system has provided a compelling biological rationale for the cognitive impairments associated with chronic sleep deprivation and for the epidemiological associations between poor sleep and increased dementia risk. The brain may need adequate sleep specifically to clear the metabolic byproducts of waking neural activity before they accumulate to pathological levels. The autonomic nervous system maintains involuntary physiological functions through two complementary divisions: the sympathetic nervous system, which increases heart rate, blood pressure, and metabolic rate in preparation for physical activity or stress, the fight or flight response, and the parasympathetic nervous system, which promotes rest, digestion, and recovery, the rest and digest state. These two divisions generally operate in opposition, with sympathetic activation suppressing parasympathetic function and vice versa, though the relationship is more nuanced than simple antagonism in many physiological contexts. Heart rate variability, the variation in time intervals between successive heartbeats, is a widely used measure of autonomic nervous system function. High variability, reflecting flexible and responsive autonomic control, is generally associated with better health outcomes, while low variability, indicating more rigid autonomic control, is associated with increased cardiovascular risk. The vagus nerve, the main parasympathetic outflow to the viscera, plays a central role in regulating heart rate, gut function, immune activity, and the inflammatory response, and its stimulation, either naturally through slow, deep breathing or artificially through vagus nerve stimulation devices, has been found to have wide-ranging physiological and psychological effects. The lymphatic system is a parallel circulation that runs throughout the body alongside the blood vascular system, collecting the fluid that leaks from blood capillaries into the tissue spaces and returning it to the bloodstream via the thoracic duct, which empties into the venous circulation near the heart. Unlike the blood circulatory system, the lymphatic system has no pump. Lymph flow is driven by the compression of lymphatic vessels by surrounding muscle contractions, breathing movements, and arterial pulsations. The lymphatic system also serves as a highway for immune cells, transporting lymphocytes and other immune cells between the lymph nodes, small oval structures scattered throughout the body that filter lymph and concentrate immune responses, and the tissues. The lymph nodes are the sites where antigen-presenting cells display fragments of pathogens to lymphocytes, initiating the adaptive immune response that produces antibodies and cytotoxic T cells specific to the particular pathogen encountered. The endocrine system communicates through hormones, chemical messengers secreted into the bloodstream that travel to target organs and cells, where they bind to specific receptors and trigger specific responses. The major endocrine glands include the pituitary, which secretes hormones that regulate growth, reproduction, stress response, and the function of other endocrine glands, the thyroid, which regulates metabolic rate, the adrenals, which secrete cortisol, epinephrine, and other stress-related hormones, the gonads, which produce sex hormones, and the pancreas, discussed earlier. The endocrine system is a communication network of extraordinary subtlety, with hormones acting over timescales ranging from seconds, in the case of epinephrine, to days or weeks, in the case of thyroid hormone, and with effects ranging from immediate changes in heart rate to long-term changes in gene expression patterns that persist for weeks after the hormonal signal has dissipated. The brain's reward system, centered on the mesolimbic dopamine pathway, plays a central role in motivation, learning, and the reinforcement of behavior. When an organism encounters something rewarding, food, social connection, achievement, the ventral tegmental area in the midbrain releases dopamine into the nucleus accumbens and the prefrontal cortex, producing the subjective experience of pleasure and reinforcing the behavior that led to the reward. Crucially, the dopamine system responds not simply to rewards themselves, but to signals that predict rewards. The anticipation of reward produces strong dopamine release, and more strongly to unexpected rewards than to expected ones. This prediction error signaling makes the dopamine system a powerful learning machine, reinforcing behaviors that reliably produce good outcomes and adjusting predictions when actual outcomes differ from expectations. The dopamine system is the primary target of most addictive substances and behaviors, which produce dopamine release at levels far exceeding those produced by natural rewards, overwhelming the normal regulation of the system. The cerebellum, the cauliflower-shaped structure at the back of the brain, contains approximately 80% of the brain's total neuron count despite representing only about 10% of the brain's volume, because its neurons are extremely small and densely packed. Traditionally understood to coordinate movement and maintain balance, the cerebellum has more recently been found to be involved in a range of cognitive and emotional functions including attention, working memory, language processing, and the regulation of emotional responses. The cerebellum appears to function as a general-purpose predictor, comparing actual sensory feedback with predicted sensory feedback generated by an internal model of the body and generating error signals when the two diverge. In the motor domain, this predictive function allows the cerebellum to compensate for delays in sensory feedback by calculating what the sensory outcome of a movement should be before it can actually be measured, enabling the smooth, precise movements that cerebellar damage impairs. The hippocampus, a paired structure in the medial temporal lobe of each cerebral hemisphere, is critical for the formation of new declarative memories, memories for facts and events, and for spatial navigation. Patients with damage to both hippocampi, such as the famous patient HM who had his hippocampi removed to control severe epilepsy in 1953, are unable to form new long-term memories, but retain memories formed before the damage and retain procedural memories and other forms of implicit learning. Research on hippocampal function has found that hippocampal neurons called place cells fire in highly specific spatial locations, and that networks of place cells together map the environment the animal is navigating. In humans, evidence from brain imaging studies and from the famous study of London taxi drivers, who showed enlarged posterior hippocampi compared to non-taxi drivers, correlating with years of experience navigating the complex streets of London, supports the role of the hippocampus in spatial navigation and in the formation of cognitive maps. The prefrontal cortex, the large region of cerebral cortex at the front of the brain, is associated with higher cognitive functions including planning, working memory, decision-making, impulse control, and the regulation of emotional responses. It is the most recently evolved part of the brain in terms of relative size, occupying a larger fraction of the total brain volume in humans than in any other species. The prefrontal cortex continues to develop and mature well into the mid-20s, with myelin deposition, the insulation of nerve fibers that speeds neural transmission, continuing in prefrontal regions until approximately age 25. This late maturation of the prefrontal cortex is thought to underlie some of the characteristic differences between adolescent and adult cognition and behavior, including greater risk-taking, greater emotional reactivity, and less stable long-term planning in adolescents compared to adults. The prefrontal cortex exerts top-down regulatory control over the amygdala and other limbic structures, reducing emotional reactivity in contexts where this is appropriate, and this regulatory capacity strengthens through adolescence and into early adulthood as the prefrontal cortex continues to mature. The amygdala, a pair of almond-shaped structures in the medial temporal lobe, plays a central role in processing emotionally significant stimuli, particularly threatening ones. Lesions of the amygdala produce a distinctive syndrome, including reduced fear responses, impaired recognition of fearful faces, and in some cases inappropriate approach behavior toward potentially dangerous stimuli. The amygdala processes sensory information about potentially threatening stimuli very rapidly, in some cases before the cortex has fully analyzed the stimulus, through a direct pathway from the thalamus to the amygdala that bypasses cortical processing, allowing a rapid preparatory response to potential threats before conscious awareness. This thalamo-amygdala shortcut is thought to underlie some aspects of the rapid, automatic fear responses that can occur before any conscious recognition of a threat, and it may contribute to the formation of fear associations that are acquired rapidly and are resistant to extinction. The body's stress response system, centered on the hypothalamic pituitary adrenal axis, is designed for acute, short-duration activation. When the hypothalamus detects a stressor, it releases corticotropin-releasing hormone, which stimulates the pituitary to release adrenocorticotropic hormone, which stimulates the adrenal cortex to release cortisol. Cortisol mobilizes energy by stimulating glucose release from the liver, enhances cardiovascular function, suppresses non-essential functions including digestion, reproduction, and immune activity, and sharpens attention and memory for the stressful situation. These effects are adaptive in the short term, preparing the body for the demands of an acute challenge. The problem arises when the stressor is chronic, when cortisol is elevated persistently rather than in short bursts, because the same suppression of digestion, reproduction, immune activity, and repair processes that is adaptive in the short term becomes pathological when sustained. Chronic stress is associated with cardiovascular disease, metabolic disruption, immune dysfunction, cognitive impairment, and reduced bone density, all reflecting the long-term costs of a system designed for acute, temporary activation being chronically activated in the absence of physical challenge. The process of wound healing is a beautifully orchestrated four-phase process involving inflammatory, proliferative, and remodeling stages. When tissue is injured, the immediate response is hemostasis. Bleeding stops through the constriction of blood vessels and the formation of a platelet plug, followed by blood clotting. The inflammatory phase follows, with immune cells migrating to the wound site, clearing bacteria and debris, and releasing signaling molecules that coordinate subsequent repair. The proliferative phase involves the migration and division of fibroblasts, cells that produce collagen, along with the formation of new blood vessels, the proliferation of epithelial cells across the wound surface, and the contraction of the wound by myofibroblasts. The final remodeling phase, which can last for months to years, involves the gradual reorganization of collagen fibers to increase the strength and organization of the scar tissue. The end result of this process is scar tissue that can achieve approximately 80% of the tensile strength of the original tissue, though it lacks the full complexity and flexibility of uninjured skin. The tongue is not divided into discrete taste zones for different tastes, despite the persistent myth, derived from a misinterpretation of a 19th century paper, that sweet taste is detected at the tip, salty and sour at the sides, and bitter at the back. In reality, the approximately 10,000 taste buds distributed across the tongue each contain receptor cells sensitive to all 5 basic tastes: sweet, sour, salty, bitter, and umami, with some variation in sensitivity across regions, but no absolute zonation. Taste receptor cells also respond to fat, starch-derived glucose polymers that may constitute a 6th taste category, and chemical irritants including capsaicin from chili peppers, which activates pain receptors rather than taste receptors. The experience of flavor is not taste alone. Approximately 80% of what we perceive as taste is actually smell, processed through the olfactory system as volatile compounds released during chewing reach the olfactory epithelium via the retronasal route, which is why food seems tasteless when the sense of smell is blocked by a head cold. The human gut, if stretched out flat, would have a surface area of approximately 30 to 40 square meters, roughly the size of a small studio apartment. This enormous surface area is created by the hierarchical folding of the intestinal wall at multiple scales. The gross folds of the intestinal wall, the finger-like projections called villi that cover the inner surface, and the even finer projections called microvilli on the surface of each villus cell, which together create a brush-like surface called the brush border. This amplification of surface area, a factor of approximately 600 compared to a simple cylinder of the same length and diameter, is essential for efficient absorption of nutrients from the small intestine, which in an adult is approximately 7 meters long. The cells lining the intestinal villi turn over every 3 to 5 days, among the fastest cell renewal rates in the body, which is thought to be an adaptation to the mechanical and chemical stresses of the digestive environment and to the constant exposure to potentially harmful microorganisms in the gut contents. The human hand is one of the most mechanically complex structures in the body, containing 27 bones, 29 joints, over 30 muscles, some of which are intrinsic to the hand, and some of which are in the forearm and act on the hand through long tendons, and thousands of sensory receptors providing precise information about touch, pressure, vibration, and temperature. The thumb, whose ability to oppose the other digits allows the precision grip that is fundamental to human tool use and manipulation, is controlled by 9 separate muscles and has a range of motion that exceeds that of any other finger. The fingertips contain some of the highest densities of sensory receptors in the body, approximately 2,000 Meissner's corpuscles and Merkel's discs per square centimeter, giving the fingertips the ability to detect surface features with a spatial resolution of approximately 2 millimeters, the basis of Braille reading by trained individuals. The combination of motor precision and sensory richness in the human hand is one of the anatomical foundations of human technological capacity. The female reproductive cycle is an extraordinarily coordinated sequence of hormonal events involving the hypothalamus, pituitary gland, and ovaries that, over approximately 28 days, prepares the uterus for potential implantation of an embryo, releases an egg at ovulation, and either supports or terminates the preparation depending on whether fertilization occurs. The cycle is driven by the interacting positive and negative feedback effects of estrogen and progesterone on the hypothalamic pituitary axis, producing a precise hormonal sequence that coordinates follicular development, ovulation, and luteal phase support of a potential pregnancy. The entire cycle involves changes not only in reproductive organs, but in the cardiovascular system, the immune system, mood and cognition, and metabolism, reflecting the systemic hormonal effects of the cycle hormones and illustrating how interconnected the body systems are even in processes that might appear to be organ-specific. Bones are approximately 65% mineral, primarily hydroxyapatite, a calcium phosphate crystal, and 35% organic matrix, primarily collagen. This composite structure is what gives bone its combination of strength and flexibility. The mineral component provides compressive strength, resisting crushing, while the organic matrix provides tensile strength and flexibility, resisting bending and fracturing. Bone that has lost its organic matrix, as in some disease states, becomes brittle and fractures easily, while bone that has lost its mineral, as in rickets or osteomalacia, becomes soft and deformable. The combination of these two very different material properties in a single structure, through through the hierarchical organization of collagen fibers and mineral crystals at the molecular level, produces a mechanical performance that exceeds what either component alone could achieve, and that engineers have long studied as a model for the design of composite materials. The human body produces approximately 25 million new red blood cells per second, replacing approximately 1% of the total red blood cell population daily. Red blood cells are produced in the red bone marrow of flat bones, including the sternum, pelvis, and vertebrae, and in the proximal ends of the long bones of the limbs. The process of red blood cell production, erythropoiesis, is precisely regulated by the hormone erythropoietin, produced primarily by the kidneys in response to low oxygen levels in the blood, ensuring that red blood cell production increases when oxygen delivery is inadequate and decreases when oxygen delivery is sufficient. When red blood cells complete their approximately 120-day lifespan, they are removed from circulation by the spleen and liver, which break down the hemoglobin and recycle its components. The iron is recovered and returned to the bone marrow for use in new hemoglobin molecules, while the porphyrin ring of the heme group is converted to bilirubin and excreted in bile. Mitochondria are often described as the power stations of the cell, but that phrase barely captures how strange they are. They are tiny structures inside nearly every human cell that convert energy from food into ATP, the molecule cells use as their immediate energy currency. Each mitochondrion has its own DNA, separate from the DNA in the cell nucleus, because mitochondria descend from ancient bacteria that entered into partnership with the ancestors of complex cells more than 1 billion years ago. That event, one cell living inside another and becoming indispensable, was one of the most important evolutionary transitions in the history of life. Without mitochondria, complex multicellular animals like humans almost certainly could not exist. The energy demands of the brain, the muscles, the heart, the liver, and the immune system are too large to be powered by simpler cellular chemistry alone. The ATP produced by mitochondria is not stored for long. The body uses and regenerates it continuously. At any given moment, the total amount of ATP present in the body is only a small fraction of what is used over the course of a day, because each molecule is recycled again and again. The human body may turn over roughly its own body weight in ATP every day, depending on activity level, because every muscle contraction, every nerve impulse, every transport of ions across a membrane, every step in protein synthesis, and every act of cellular maintenance requires energy. The body is therefore not powered by a static reserve. It is powered by a river of chemical regeneration, a constant cycling of energy through molecules too small to see, happening trillions of times per second. Every living cell maintains an electrical difference across its membrane. This is called the membrane potential, and it exists because ions, charged atoms such as sodium, potassium, calcium, and chloride, are distributed unequally between the inside and outside of the cell. Cells spend a significant portion of their energy maintaining these gradients. The sodium-potassium pump, one of the most important proteins in the body, uses ATP to move sodium ions out of the cell and potassium ions into it, preserving the conditions that make electrical signaling possible. In neurons and muscle cells, tiny changes in this electrical balance become action potentials, rapid waves of electrical change that carry information or trigger contraction. Consciousness, heartbeat, movement, reflex, and sensation all depend on the quiet maintenance of ion gradients. The cell membrane is not just a boundary. It is an active, selective, dynamic surface where the cell negotiates with the outside world. Embedded in the membrane are receptors that detect hormones, neurotransmitters, immune signals, nutrients, and warning molecules. There are channels that open and close to allow specific ions to pass. There are transporters that bring glucose, amino acids, and other molecules into the cell. There are adhesion proteins that allow cells to attach to one another and organize into tissues. The membrane is thin enough to be invisible under an ordinary microscope, yet it is the place where much of the body's communication begins. A hormone released in the blood may affect a distant organ only because a membrane receptor recognizes it and translates that external signal into an internal response. Genes are not simply switched on or off in a permanent way. A liver cell, a neuron, a skin cell, and a muscle cell contain essentially the same DNA, but they behave differently because different sets of genes are active in each one. This control is achieved through gene regulation, molecular systems that determine when a gene is read, how strongly it is expressed, and how long the resulting RNA or protein persists. Epigenetic marks, including chemical modifications to DNA and to the histone proteins around which DNA is wrapped, help regulate these patterns. They do not change the DNA sequence itself, but they influence how accessible different regions of the genome are to the cellular machinery that reads genes. Development is, in large part, the progressive establishment of stable patterns of gene expression in cells that all began from the same fertilized egg. The body also maintains itself through controlled cell death. This process, called apoptosis, is not a failure or an accident. It is a necessary part of life. During development, apoptosis sculpts the fingers and toes by removing the cells between them. In the immune system, it eliminates immune cells that might attack the body's own tissues. In adult tissues, it removes cells that are damaged, infected, or no longer needed. A cell undergoing apoptosis does not burst open and spill its contents in a damaging way. It dismantles itself carefully, packaging its components into small membrane-bound fragments that neighboring cells or immune cells can clear away. The body is continuously building, renewing, and removing itself with astonishing discipline. Healthy life depends not only on cells dividing, but on cells knowing when to disappear. Stem cells are the reason many tissues can renew. A stem cell is defined by two properties. It can make more copies of itself, and it can produce more specialized descendant cells. In the bone marrow, blood-forming stem cells generate red blood cells, white blood cells, and platelets throughout life. In the intestine, stem cells near the base of crypts continuously produce new cells that migrate upward to replace the lining of the gut every few days. In the skin, stem cells replenish the epidermis as outer cells are shed. The body does not regenerate all tissues equally. The lining of the gut and the blood renew rapidly. The heart regenerates poorly. Many neurons are meant to last a lifetime. This uneven capacity for repair is one of the major reasons different organs age and recover differently. The human skeleton is often imagined as the body's frame, but it is also a mineral bank, a blood factory, and an endocrine organ. Inside the marrow cavities of many bones, blood cells are produced continuously. The mineral matrix of bone stores calcium and phosphate, releasing or absorbing them to help maintain the precise blood levels required for nerve function, muscle contraction, and cellular signaling. Bone cells also release hormones, including osteocalcin, which appears to influence metabolism and other physiological processes. Bone is therefore not dead architecture. It senses mechanical load, communicates chemically with other organs, repairs microdamage, and remodels itself according to use. A person who walks, lifts, or moves regularly gives their bones signals to maintain strength. A body deprived of mechanical stress begins to dissolve part of its own skeleton. Cartilage is one of the body's quiet engineering achievements. It covers the ends of bones within joints, creating a smooth, low-friction surface that allows movement under load. Healthy articular cartilage has no blood vessels and no nerves, which is why it can function as a nearly frictionless bearing surface, but also why it heals poorly when damaged. Its structure is maintained by chondrocyte, specialized cells embedded in a dense extracellular matrix of collagen and proteoglycans that trap water and resist compression. In a knee, hip, or shoulder, cartilage allows bones to glide against one another through millions of cycles of movement. When cartilage breaks down, as in osteoarthritis, the loss of that smooth surface transforms ordinary movement into inflammation, pain, and mechanical wear. The ease of movement that feels natural is actually the result of an intricate biological material science. Tendons and ligaments reveal another kind of biological design. Tendons connect muscle to bone, transmitting the force generated by muscle contraction into movement of the skeleton. Ligaments connect bone to bone, stabilizing joints and limiting movement that would damage them. Both are rich in collagen, arranged in strong parallel fibers that can withstand enormous tensile force. Tendons also store and release elastic energy. The Achilles tendon, for example, stretches slightly when the foot strikes the ground and recoils as the body pushes forward, making walking and running more efficient. This spring-like function reduces the metabolic cost of movement. The body is not only powered by muscles, it also uses elastic tissues to recycle energy from one step into the next. The human face contains more than 40 muscles, many of which are specialized not for chewing or survival in the narrow mechanical sense, but for expression. Smiling, frowning, squinting, raising the eyebrows, tightening the lips, widening the eyes. These movements communicate emotional information quickly and often involuntarily. The facial nerve carries motor signals from the brain to these muscles, and tiny differences in contraction produce expressions that other humans can read with remarkable speed. The face is therefore a social organ as much as an anatomical one. It displays fear before words can explain it, warmth before speech begins, fatigue before a person admits it, and pain even when the conscious mind tries to hide it. Much of human social life depends on muscles so small and familiar that we rarely think of them as physiology. The human voice is produced by a delicate collaboration between breath, vibration, resonance, and articulation. Air from the lungs passes through the larynx, where the vocal folds vibrate hundreds of times per second, producing a raw, buzzing sound. The shape of the throat, mouth, nasal passages, tongue, lips, and jaw then filters and shapes that sound into vowels, consonants, tone, and speech. The vocal folds are small, soft structures, but they can produce an enormous range of pitch and intensity through precise muscular control. Speech requires coordination between the respiratory system, laryngeal muscles, tongue, lips, jaw, auditory feedback, and brain regions involved in language and motor planning. A spoken sentence is therefore not just a thought turned into sound. It is a high-speed motor performance involving the whole upper airway. The diaphragm is the primary muscle of breathing. It forms a dome-shaped sheet beneath the lungs, separating the chest cavity from the abdominal cavity. When it contracts, it flattens downward, increasing the volume of the chest and lowering the pressure inside the lungs, causing air to flow inward. When it relaxes, the elastic recoil of the lungs and chest wall pushes air back out. Most of the time, breathing happens automatically, regulated by centers in the brainstem that monitor carbon dioxide levels in the blood. Yet breathing is also unusual because it can be voluntarily controlled. You can hold your breath, speak, sing, sigh, hum, or slow your breathing intentionally. This makes breathing one of the few bridges between automatic physiology and conscious control, which is why slow breathing can influence heart rate, stress state, and the balance of the autonomic nervous system. The lungs are lined with a substance called surfactant, produced by specialized cells in the alveoli. Surfactant reduces surface tension, preventing the tiny air sacs from collapsing after each breath. Without it, the force required to reopen the alveoli would be enormous, and breathing would become exhausting or impossible. Premature infants may struggle to breathe because their lungs have not yet produced enough surfactant, which is why surfactant therapy became one of the major advances in neonatal medicine. Every quiet breath depends on this molecular film spread across millions of microscopic air sacs. The simplicity of breathing hides the fact that the lungs are constantly managing fluid surfaces, pressure gradients, elastic recoil, and chemical exchange with exquisite precision. The blood clotting system is one of the body's most finely balanced mechanisms. If blood did not clot, even small injuries could become dangerous. If blood clotted too easily, vessels could become blocked, causing strokes, heart attacks, or pulmonary embolisms. When a vessel is injured, platelets adhere to the damaged surface, change shape, and release signals that recruit more platelets. At the same time, a cascade of clotting proteins in the blood activates in sequence, ultimately producing fibrin, a strong protein mesh that stabilizes the clot. Later, once repair is underway, another system begins dissolving the clot to restore normal flow. Hemostasis is therefore not a simple plug. It is a temporary biological construction project, assembled quickly, held only as long as needed, and then dismantled. Human blood types are determined by molecules on the surface of red blood cells. In the ABO system, type A blood has A antigens, type B has B antigens, type AB has both, and type O has neither. The immune system naturally produces antibodies against the ABO antigens it does not have, which is why incompatible blood transfusions can be dangerous. The Rh system, especially the Rhd antigen, is another major blood group factor. Blood type is often discussed casually, but it represents an important example of how individual variation at the molecular surface of cells can have major medical consequences. Safe transfusion medicine depends on recognizing that blood from one person is not automatically interchangeable with blood from another. Even a life-saving tissue can become dangerous if the immune system reads it as foreign. The spleen is an often overlooked organ tucked beneath the left side of the rib cage, but it performs several essential functions. It filters the blood, removing old or damaged red blood cells and recycling their components. It stores platelets and certain immune cells. It helps detect blood-borne pathogens and mount immune responses against them. The spleen is especially important in defending against certain encapsulated bacteria, which can evade other immune mechanisms unless properly tagged and cleared. A person can survive without a spleen, but their risk of severe infection from particular bacteria increases, which is why vaccination and medical precautions become important after splenectomy. The spleen is like a quiet checkpoint in the bloodstream, inspecting cells, clearing debris, and helping maintain immune readiness. The thymus is most active in childhood and is essential for the development of T cells, a major branch of the adaptive immune system. Immature T cells travel to the thymus, where they undergo a strict selection process. They must be able to recognize the body's own antigen-presenting molecules, but they must not react too strongly against the body's own tissues. Cells that fail these tests die. This process is one of the reasons the immune system can defend against pathogens without usually attacking the self. The thymus gradually shrinks after puberty, replaced partly by fat, but the immune education that occurred there continues to shape immune function throughout life. A small organ most people rarely think about help train the cellular system that distinguishes protection from autoimmunity. Inflammation is often treated as something purely negative, but it is one of the body's central repair and defense mechanisms. When tissue is injured or infected, blood vessels dilate, local blood flow increases, immune cells move into the tissue, and chemical signals coordinate the response. The classic signs, redness, heat, swelling, pain, and loss of function, are not random symptoms. They reflect increased blood flow, fluid movement, immune activity, and protective sensitivity. Acute inflammation helps isolate threats, clear damaged material, and begin repair. Chronic inflammation is different. When inflammatory signaling remains active for weeks, months, or years, it can damage tissues and contribute to disease. The same system that protects the body in the short term can harm it when it fails to turn off. Fever is another defense mechanism that is often misunderstood. When the immune system detects infection, it releases signaling molecules called pyrogens that act on the hypothalamus, raising the body's temperature set point. The person feels cold because the current body temperature is now below the new set point, so shivering and vasoconstriction help raise temperature. A moderate fever can slow the replication of some pathogens and enhance certain immune functions. Fever is therefore not simply a malfunction. It is an active, regulated state. That said, very high or prolonged fevers can become dangerous, and fever must be interpreted in context. But the basic idea remains remarkable. The body can deliberately change its thermal environment as part of its strategy for survival. Antibodies are Y-shaped proteins produced by B cells, and their specificity is one of the marvels of the adaptive immune system. Each antibody binds to a particular molecular shape, known as an antigen. When the immune system encounters a pathogen, B cells that happen to make antibodies matching that pathogen are activated, multiply, and refine their antibodies through a process called affinity maturation. Some become plasma cells that produce large quantities of antibody. Others become memory B cells that persist long after the infection is gone. This memory is why a second encounter with the same pathogen can be met faster and more effectively than the first. Vaccines work by safely training this memory system, allowing the immune system to rehearse recognition without requiring the full danger of the disease itself. The complement system is a set of proteins in the blood that acts like a biochemical alarm and attack network. When activated, complement proteins can tag microbes for destruction, attract immune cells to the site of infection, amplify inflammation, and in some cases form pores in microbial membranes that directly kill the invader. The system operates through cascades, where one activated protein activates the next, producing rapid amplification from a small initial signal. Because it is powerful, complement must be tightly regulated. If it attacks the body's own cells, serious disease can result. It is one of the older parts of immunity, evolutionarily ancient compared with antibodies, and it shows how much of defense happens below conscious awareness, through chemical recognition systems moving silently through the blood. The brain is protected by multiple layers. The skull provides hard physical protection. The meninges, three membranes called the dura mater, arachnoid mater, and pia mater, surround the brain and spinal cord. Cerebrospinal fluid cushions the nervous tissue and helps maintain its chemical environment. The blood-brain barrier, formed by tightly sealed endothelial cells and brain capillaries along with support from astrocytes and parasites, carefully controls which substances can pass from blood into brain tissue. This barrier protects the brain from toxins and pathogens, but it also makes treating many brain diseases difficult because many medications cannot cross it easily. The brain is therefore both privileged and vulnerable, protected from much of the body's chemical turbulence, yet difficult to reach when something goes wrong. Glial cells were once thought of as merely support cells for neurons, but they are now recognized as active participants in brain function. Astrocytes help regulate the chemical environment around synapses, support the blood-brain barrier, and influence synaptic signaling. Oligodendrocytes produce myelin, the insulating sheath that allows rapid signal conduction along axons in the central nervous system. Microglia are the brain's resident immune cells, constantly surveying the tissue for damage, infection, and abnormal activity. During development, microglia help prune synapses, removing connections that are no longer needed. The brain is therefore not just a network of neurons. It is an ecosystem of cell types whose cooperation makes thought possible. The cells once considered background are part of the foreground of consciousness. Myelin is the fatty insulation around many nerve fibers, and it dramatically increases the speed of electrical signaling. In an unmyelinated axon, an electrical impulse must propagate continuously along the membrane. In a myelinated axon, the impulse effectively jumps between gaps in the myelin called nodes of Ranvier, a process called saltatory conduction. This allows signals to travel much faster and with less energy use. Myelination continues throughout childhood and adolescence and into early adulthood, especially in brain networks involved in complex cognition and self-control. Diseases that damage myelin, such as multiple sclerosis, show how essential this insulation is. When myelin is lost, signals slow, distort, or fail, producing symptoms that can affect movement, sensation, vision, and cognition. Speed of thought depends partly on biological insulation. Memory is not stored in the brain as a simple recording. It is reconstructed through patterns of neural activity distributed across many networks. The hippocampus helps bind elements of an experience, place, time, emotion, people, sensory details into a retrievable episode. Over time, with sleep and repeated reactivation, aspects of memory become increasingly integrated into the cortex. Each act of remembering can also modify the memory, because retrieval makes memory traces temporarily flexible before they are stored again, a process called reconsolidation. This is why memory is both powerful and imperfect. It allows the past to guide the present, but it is not a fixed archive. It is a living system shaped by attention, emotion, sleep, meaning, and later experience. Pain is not simply a measure of tissue damage. It is an experience constructed by the nervous system based on signals from the body, context, expectation, memory, emotion, and threat assessment. Nociceptors detect potentially damaging stimuli such as heat, pressure, or chemical irritation and send signals to the spinal cord and brain. But the brain can amplify or dampen those signals. A soldier injured in battle may feel surprisingly little pain until danger has passed. A minor injury can feel intense if it is frightening or uncertain. Chronic pain can persist even after tissue healing because the nervous system itself becomes sensitized. Pain is therefore protective, but it can also become maladaptive. It is the body's alarm system, and like any alarm system, it can be life-saving when calibrated well and devastating when stuck on. Proprioception is the sense of where the body is in space. It allows you to touch your nose with your eyes closed, walk without watching your feet, and know whether your elbow is bent without looking. This sense depends on receptors in muscles, tendons, and joints that detect stretch, tension, and position. The brain integrates this information with signals from the inner ear and vision to maintain posture and coordinate movement. Proprioception is usually invisible to awareness because it works so smoothly. When it is disrupted, movement becomes profoundly difficult. The body must be watched constantly to be controlled. Ordinary grace, reaching for a cup, turning in bed, walking through a dark room, depends on a hidden sensory system constantly reporting the body's shape to itself. The vestibular system, located in the inner ear, detects head movement and orientation relative to gravity. It contains 3 semicircular canals, arranged roughly at right angles, that detect rotational movement, and 2 otolith organs that detect linear acceleration and head tilt. Tiny hair cells inside these structures convert mechanical movement of fluid and small calcium carbonate crystals into electrical signals sent to the brain. The vestibular system stabilizes vision during head movement through the vestibulo-ocular reflex, which moves the eyes in the opposite direction of head motion to keep the visual world steady. It also contributes to balance, posture, and the sense of bodily orientation. Dizziness and vertigo reveal how deeply the brain relies on this system. When vestibular signals conflict with vision or proprioception, the world itself seems to move. The sense of touch is actually a family of senses. Different receptors in the skin detect light pressure, deep pressure, vibration, stretch, temperature, itch, and pain. Meissner's corpuscles detect light, fluttering touch. Pacinian corpuscles detect vibration. Merkel cells detect sustained pressure and fine texture. Ruffini endings detect skin stretch. Free nerve endings detect pain, temperature, and itch. These signals travel through different pathways to the spinal cord and brain, where they are mapped in the somatosensory cortex. The hands and lips occupy disproportionately large areas of this map because their sensory resolution is so high. The body does not represent itself according to physical size. It represents itself according to sensory importance. Itch is one of the stranger sensations the body produces. It is related to pain but distinct from it, carried by specific nerve fibers and processed through overlapping but separate pathways. Itch likely evolved as a protective signal to remove insects, parasites, or irritating substances from the skin. Scratching temporarily relieves itch by activating pain and touch signals that suppress itch transmission in the spinal cord. But scratching also damages the skin and can worsen inflammation, producing the itch-scratch cycle seen in many skin conditions. The fact that seeing someone else scratch can make a person feel itchy shows that itch is not purely local. It can be socially and visually triggered, revealing once again how bodily sensation is shaped by the brain's interpretation of context. The sense of balance between hunger and fullness is controlled by a complex conversation between the gut, fat tissue, pancreas, liver, and brain. Hormones such as ghrelin, produced mainly by the stomach, increase hunger. Leptin, produced by fat cells, signals long-term energy stores. Insulin reflects nutrient availability. Peptide YY, GLP-1, and cholecystokinin released from the gut contribute to satiety after eating. These signals converge in the hypothalamus and other brain regions that regulate appetite, reward, and energy expenditure. Eating is therefore not controlled by willpower alone. It is regulated by a deep physiological system evolved to defend energy balance in environments where food availability was unpredictable. Modern food environments can overwhelm this ancient system with constant availability, high palatability, and dense calories. The stomach protects itself from its own acid through a mucus bicarbonate barrier. Gastric acid is strong enough to denature proteins and kill many microbes entering with food. The stomach lining secretes hydrochloric acid into the lumen, while simultaneously protecting its own cells with mucus rich in bicarbonate, which neutralizes acid at the surface. Specialized cells produce enzymes, hormones, and acid in coordinated patterns regulated by nerves and hormones. If the protective barrier is disrupted, acid can damage the lining, contributing to ulcers. The discovery that many stomach ulcers are caused by the bacterium Helicobacter pylori, rather than simply stress or excess acid, transformed gastroenterology and won a Nobel Prize. Even in the acidic stomach, life found a niche. Peristalsis is the coordinated wave of muscular contraction that moves food through the digestive tract. It is controlled largely by the enteric nervous system, with modulation from the autonomic nervous system and hormones. After swallowing, the esophagus pushes food downward even if a person is upside down, because movement is driven by muscular waves rather than gravity alone. In the intestines, peristalsis mixes and moves contents, allowing digestion and absorption to occur in sequence. The digestive system is not just a tube. It is a rhythmic, responsive organ system that senses stretch, chemical composition, microbial signals, and nervous input, adjusting speed and secretion according to what has been eaten and what the body needs. The colon reclaims water and electrolytes from material that remains after digestion and absorption in the small intestine. It is also a dense microbial ecosystem where bacteria ferment fibers and resistant starches that human enzymes cannot digest. This fermentation produces short-chain fatty acids, including acetate, propionate, and butyrate. Butyrate is especially important because it nourishes the cells lining the colon and has anti-inflammatory effects. The relationship between human host and gut microbes is therefore a metabolic partnership. We feed them complex carbohydrates. They return molecules that influence gut health, immune function, and possibly even distant organs through circulation and signaling. Digestion does not end with human enzymes. It continues through microbial chemistry. The liver's detoxification work occurs in phases. In broad terms, phase 1 enzymes, many belonging to the cytochrome P450 family, chemically modify substances such as drugs, hormones, and environmental chemicals. Phase 2 pathways then attach other molecules to make these substances more water-soluble, allowing them to be excreted in bile or urine. This system is powerful but not magical. Some compounds become temporarily more reactive during processing, and liver capacity can be overwhelmed by high doses of toxins such as acetaminophen. The liver also handles the ordinary chemistry of life, breaking down hormones after their signals are complete, managing ammonia produced by protein metabolism, and converting it into urea for safe excretion. It is the body's great chemical mediator between usefulness and danger. The kidneys do more than make urine. They regulate blood pressure, blood acidity, electrolyte balance, and red blood cell production. They help maintain the pH of the blood within a narrow range, because even small deviations in acidity can disrupt protein structure, enzyme function, and nerve signaling. They control sodium and water balance in coordination with hormones such as aldosterone and antidiuretic hormone. They activate vitamin D into its hormonally active form, which helps regulate calcium and bone metabolism. The kidneys are therefore not waste filters in the crude sense. They are precision regulators of the body's internal ocean, maintaining the chemical composition of blood so that every cell can function in a stable environment despite constant external change. The endocrine system often works through feedback loops. The hypothalamus releases signals to the pituitary gland. The pituitary releases hormones that act on distant glands, and those glands release hormones that feed back to the brain and pituitary to regulate further release. The thyroid axis is a classic example. The hypothalamus signals the pituitary releases thyroid-stimulating hormone, and the thyroid produces thyroid hormones that influence metabolic rate throughout the body. When thyroid hormone levels rise, they suppress further signaling from the hypothalamus and pituitary. This feedback keeps the system stable. Hormones are not simply released into the body at random. They are governed by loops, thresholds, rhythms, and sensitivity changes that maintain balance over time. The thyroid gland, located in the neck, has effects on nearly every tissue. Its hormones increase metabolic rate, influence body temperature, affect heart rate, support brain development, and regulate how cells use energy. Too little thyroid hormone can cause fatigue, cold intolerance, slowed heart rate, weight gain, constipation, and mental slowing. Too much can cause heat intolerance, weight loss, tremor, anxiety, rapid heart rate, and sleep disturbance. The same hormone system, shifted in one direction or another, changes the entire bodily state. The thyroid demonstrates how a small organ can tune the tempo of the whole organism. influencing whether the body feels slowed, steady, or accelerated. The adrenal glands sit above the kidneys and contain two functionally different regions. The adrenal cortex produces steroid hormones, including cortisol, aldosterone, and androgens. The adrenal medulla produces epinephrine and norepinephrine, rapidly released during acute stress. When a person is startled, frightened, or physically challenged, the adrenal medulla can flood the bloodstream with signals that increase heart rate, dilate airways, mobilize glucose, and redirect blood flow toward muscles. The adrenal cortex works on a slower timescale through cortisol and mineral balance. Together, these small glands help the body respond to danger, maintain blood pressure, regulate salt and water, and adapt to stress. Their size gives no hint of their influence. The circadian rhythm is not only about sleepiness. Nearly every organ has molecular clocks that regulate local rhythms of gene expression, metabolism, repair, and responsiveness. The central clock in the suprachiasmatic nucleus of the brain coordinates these peripheral clocks, using light as the main signal to align internal time with the external day. But feeding time, activity, temperature, and social routines also influence clocks in the liver, gut, muscle, and other tissues. This means the body is not the same at every hour. Blood pressure, hormone levels, digestion, immune activity, alertness, and body temperature all follow daily rhythms. Health depends partly on timing. A meal, a medication, or a stressor may affect the body differently depending on when it occurs. The immune system also follows daily rhythms. Certain immune cells circulate in different numbers at different times of day. Inflammatory signals rise and fall. Responses to vaccines and infections can vary according to circadian timing. This is not accidental. The body anticipates predictable environmental patterns, including activity during the day and rest at night, and coordinates immune surveillance accordingly. Sleep disruption therefore affects immunity not only by reducing rest, but by disturbing timing. A body without stable rhythms must defend, repair, digest, and regulate in a temporal environment that no longer matches its internal expectations. The skin microbiome is another invisible ecosystem. The surface of the body is home to bacteria, fungi, viruses, and mites, with different communities living in oily areas, moist areas, and dry areas. The microbes of the armpit are different from those of the forehead, which are different from those between the toes. Many of these organisms are harmless or beneficial, occupying space and resources that might otherwise be taken by pathogens, interacting with the immune system, and helping maintain skin health. The skin is therefore not a sterile barrier. It is a living borderland where human cells, microbes, oils, sweat, immune signals, and environmental exposures interact continuously. Cleanliness does not mean the absence of life. It means a healthy balance at the surface. Hair grows from follicles, miniature organs embedded in the skin. Each follicle cycles through phases. Anagen, the active growth phase, catagen, a transitional phase, and telogen, a resting phase before the hair is shed. The length of the growth phase largely determines how long hair can become. Scalp hair has a long anagen phase lasting years, while eyebrow hair has a much shorter one, which is why eyebrows do not grow like scalp hair. Hair color is produced by melanocytes in the follicle, which deposit pigment into the growing hair shaft. As melanocyte activity declines with age, hair becomes gray or white. Each hair is dead keratin by the time it emerges, but the follicle that produced it remains alive, cycling quietly beneath the skin. Nails are made of keratin, the same family of structural proteins found in hair and the outer skin. Fingernails grow from the nail matrix, a living tissue hidden beneath the cuticle. The visible nail plate is dead, compressed keratin pushed forward as new cells are produced behind it. Fingernails grow faster than toenails, and growth rates can vary with age, nutrition, circulation, hormones, and injury. Nails protect the fingertips, support fine touch by providing counterpressure to the soft pads of the fingers, and assist with grasping small objects. They are often treated as cosmetic, but anatomically they are part of what gives the human hand some of its precision. The human eye is constantly moving. Even when you think you are holding your gaze still, the eyes make tiny movements called microsaccades, drifts, and tremors. These movements prevent visual input from fading. If an image is perfectly stabilized on the retina, it begins to disappear from perception because sensory neurons adapt to unchanging stimuli. The visual system needs change to maintain experience. Larger eye movements, called saccades, occur several times per second as the eyes jump from one point of interest to another. During these jumps, the brain suppresses much of the blur that would otherwise be perceived. The stable visual world you experience is therefore not a direct feed from the eyes. It is a construction assembled from fragments, movements, predictions, and corrections. Color vision depends on 3 types of cone cells, broadly sensitive to short, medium, and long wavelengths of light. The brain compares the signals from these cone types to construct the experience of color. Color is not a property existing in the world exactly as it appears. It is the nervous system's interpretation of wavelength patterns reflected from surfaces under changing light. This is why a white page looks white in morning sun, indoor light, or shade, even though the wavelengths reaching the eye differ. The brain performs color constancy, adjusting perception according to context. Color blindness occurs when one type of cone is missing or altered, changing the comparisons the brain can make. The colors of the world are partly outside us and partly made by the visual system itself. The ear does more than detect sound. The inner ear houses both the cochlea, for hearing, and the vestibular organs, for balance. These structures share a fluid-filled architecture and hair cells that convert mechanical movement into nerve signals. Hair cells are delicate, and in humans, those in the cochlea generally do not regenerate after damage. Loud noise can bend or destroy them, leading to permanent hearing loss. This is why noise exposure accumulates across life. The ear is capable of astonishing sensitivity, but that sensitivity comes with vulnerability. Every conversation, song, footstep, alarm, whisper, and rainfall sound depends on microscopic hair cells moving in fluid in the dark of the skull. The body has a remarkable ability to predict the sensory consequences of its own actions. When you move your eyes, the visual world does not seem to smear or jump because the brain sends a copy of the motor command, an efferent copy, to sensory regions, allowing them to anticipate the movement. When you try to tickle yourself, it usually feels far less intense than when someone else does it, because the brain predicts the touch generated by your own movement and dampens the response. This predictive control helps distinguish self-generated sensations from external ones. The sense of self is partly built from the nervous system's ability to know which signals it caused. The immune system and nervous system communicate more deeply than older models of the body suggested. Immune molecules called cytokines can influence mood, sleep, appetite, and fatigue. During infection, the feeling of being sick, tired, withdrawn, achy, less hungry, less socially motivated, is not simply a side effect of damage. It is a coordinated state called sickness behavior, influenced by immune signaling to the brain. This state conserves energy, promotes rest, and reduces activity while the immune system works. The mind and immune system are not separate realms. A change in immune chemistry can alter emotion and motivation, and chronic inflammation can influence mental health. The body speaks to the brain in the language of molecules. Pregnancy is one of the most dramatic physiological transformations the human body can undergo. Blood volume expands significantly. The heart pumps more blood. The kidneys filter more. Hormone levels shift by orders of magnitude. The immune system must tolerate fetal tissue that is genetically partly distinct while still defending against infection. The placenta, a temporary organ built from both maternal and fetal tissues, mediates the exchange of oxygen, nutrients, waste products, hormones, and immune signals. It is an organ made for one relationship and one season of life, then discarded after birth. The existence of the placenta shows that the body can construct an entire temporary life support system, run it for months, and then dismantle it when its work is done. Human development begins as a single cell and unfolds through waves of division, migration, folding, specialization, and program change. In early embryonic development, cells that appear similar begin receiving different signals depending on their position. Those signals alter gene expression, and small differences become tissues, organs, axes, and body regions. The nervous system forms from a sheet of cells that folds into a tube. The heart begins beating before it has its final mature structure. Limbs emerge as buds. Fingers separate through controlled cell death. Development is not a miniature body simply growing larger. It is a sequence of transformations, each setting conditions for the next, with timing so important that small disruptions at specific stages can have lasting consequences. Puberty is a whole-body reorganization driven by the reactivation of the hypothalamic-pituitary gonadal axis. Hormonal signals stimulate the gonads to produce sex hormones, which then influence growth, bone maturation, body composition, skin, hair, reproductive organs, mood, sleep timing, and brain development. The adolescent growth spurt occurs when growth hormone, sex hormones, and bone growth plates interact. Eventually, rising sex hormone levels contribute to the closure of the growth plates, ending height increase. Puberty is therefore not just reproductive maturation. It is a coordinated shift in the entire organism, changing appearance, physiology, behavior, circadian rhythm, and social experience during a period when the brain is also still refining its control systems. Aging occurs at many levels at once. DNA accumulates damage. Telomeres, the protective caps at the ends of chromosomes, shorten in many dividing cells. Proteins become damaged or misfolded. Mitochondria become less efficient. Stem cell pools change. Inflammation tends to rise. Senescent cells, cells that no longer divide but remain metabolically active, can accumulate and secrete signals that affect neighboring tissue. None of these mechanisms alone explains aging completely. Aging is not one process, but a network of interacting processes that gradually reduce repair capacity and resilience. The body does not simply wear out like a machine. It changes its regulatory state, its cellular composition, its immune tone, and its ability to recover from stress. The body repairs DNA constantly. Every cell experiences DNA damage from normal metabolism, ultraviolet light, replication errors, and environmental exposures. Cells have multiple repair systems, each specialized for different kinds of damage. Base excision repair, nucleotide excision repair, mismatch repair, double-strand break repair. If damage is too severe, a cell may pause division, repair itself, enter senescence, or undergo apoptosis. Cancer develops when cells acquire mutations that allow uncontrolled growth, evade normal death signals, stimulate blood vessel formation, and escape immune surveillance. The fact that cancer is common reflects how many cells divide and how complex the body's regulatory systems are. The fact that cancer is not even more common reflects how much repair and surveillance happens successfully every day. The body maintains blood glucose within a narrow range because both excess and deficiency are dangerous. The brain depends heavily on glucose under ordinary conditions, and very low blood sugar can impair consciousness. High blood sugar damages blood vessels and tissues over time. After a meal, insulin signals cells to take up glucose and signals the liver and muscles to store it as glycogen. Between meals, glucagon and other hormones signal the liver to release glucose. During prolonged fasting, the body can produce ketone bodies from fat, which the brain can use as an alternative fuel. Energy regulation is therefore flexible. The body shifts between absorbed food, stored glycogen, fat metabolism, and internal glucose production according to timing and need. The human body contains several kinds of fat tissue. White fat stores energy and secretes hormones involved in appetite, metabolism, and inflammation. Brown fat burns energy to produce heat, especially in infants, who have a high need for thermoregulation and cannot shiver as effectively as adults. Brown fat contains many mitochondria and a protein called UCP1 that allows energy from fuel to be released as heat rather than captured as ATP. Adults also retain small amounts of brown and beige fat, especially in certain regions of the neck and upper torso, and cold exposure can increase its activity. Fat is therefore not inert padding. It is an endocrine and metabolic tissue, storing, signaling, insulating, cushioning, and sometimes generating heat. The body is full of rhythms shorter than a day. The heart beats in cycles. Breathing rises and falls. The stomach has slow waves of electrical activity. Hormones pulse. Growth hormone is released in bursts, especially during deep sleep. Cortisol rises before waking. Reproductive hormones are secreted in pulses that carry information through their timing, not merely their amount. A constant level of some hormones would not produce the same effect as a pulse signal. The body communicates through patterns in time. Physiology is not a flat line, but a composition of rhythms layered over one another, milliseconds in neurons, seconds in heartbeats, minutes in hormone pulses, hours in sleep cycles, days in circadian rhythms, and years in development and aging. The human body is asymmetrical in subtle but important ways. The heart sits slightly to the left. The right lung has 3 lobes while the left has 2, leaving space for the heart. The liver is mostly on the right. The stomach curves to the left. The brain has functional lateralization, with language usually more strongly represented in the left hemisphere for most people, while certain aspects of spatial attention are often more right lateralized. During embryonic development, the body establishes left-right asymmetry through molecular signals and tiny cilia that create directional fluid flow. When this process is altered, organs can develop mirrored, a condition called situs inversus. The body's apparent symmetry on the outside conceals a carefully organized internal handedness. The body has multiple barriers separating one biological compartment from another. The skin separates internal tissues from the external environment. The gut lining separates the bloodstream from the enormous microbial and chemical complexity of digested food. The blood-brain barrier protects neural tissue. The placental barrier mediates exchange between mother and fetus. The blood-testis barrier protects developing sperm from immune attack. These barriers are not walls in the absolute sense. They are selective interfaces, allowing some molecules and cells through while blocking others. Much of physiology happens at borders. Health depends on barriers being neither too closed nor too open. A leaky barrier can trigger inflammation. An overly restrictive barrier can prevent necessary exchange. The human body is not sterile, even internally in the simplistic sense once imagined. Different body sites have their own microbial ecosystems, and even tissues previously assumed to be entirely microbe-free are being re-examined with more sensitive techniques, though contamination and interpretation remain challenging in this field. What is clear is that the microbiome shapes immune development, metabolism, and barrier function from early life. Birth route, breastfeeding, antibiotics, diet, environment, pets, siblings, and geography all influence microbial colonization. The immune system learns in conversation with microbes. A body raised in total microbial absence, as shown in germ-free animal studies, develops abnormal immune and metabolic features. Human life is not solitary at the cellular scale. It is ecological. The cardiovascular system responds instantly to posture. When a person stands up, gravity pulls blood toward the legs. Sensors called baroreceptors in the carotid arteries and aorta detect the momentary drop in blood pressure and trigger reflex adjustments. Heart rate increases slightly, blood vessels constrict, and venous return is supported by muscle tone. Without this reflex, standing could cause fainting as blood flow to the brain drops. The simple act of getting out of bed requires rapid coordination between blood vessels, nerves, heart, and gravity. The body is constantly solving physics problems that conscious awareness never sees. The veins of the legs contain valves that help blood return upward toward the heart against gravity. Skeletal muscle contractions squeeze the veins, and the valves prevent blood from flowing backward. This muscle pump is especially important during walking. Long periods of immobility reduce this pumping action, which is one reason prolonged sitting or bed rest can increase the risk of blood clots in the legs. Movement is therefore part of circulation. The heart drives blood through arteries, but ordinary muscle activity helps return it through veins. The body was built for intermittent motion, and many systems work better when that motion is present. The body's oxygen transport system depends on iron atoms embedded within hemoglobin. Each hemoglobin molecule can bind 4 oxygen molecules, and each red blood cell contains hundreds of millions of hemoglobin molecules. The color of blood changes depending on oxygenation because oxygen binding alters the electronic structure of the iron-containing heme groups, changing how they absorb and reflect light. Oxygenated blood is bright red. Deoxygenated venous blood is darker, though not blue. The blue appearance of veins beneath the skin is an optical effect caused by how light penetrates and scatters through tissue. The body's colors are often physics as much as biology. The liver and muscles store glucose as glycogen, a branch polymer that can be rapidly broken down when energy is needed. Liver glycogen helps maintain blood glucose between meals, especially for the brain and other glucose-dependent tissues. Muscle glycogen is used locally by the muscle during activity. This means the body stores carbohydrate in compartments with different purposes, one for whole body blood sugar regulation, one for local movement. During sleep, fasting, exercise, or stress, the body shifts the use and replenishment of these stores according to hormonal signals. Even when you are not eating, the body is managing fuel minute by minute. The body produces heat continuously as a byproduct of metabolism. Every chemical reaction is not perfectly efficient, and the lost energy becomes heat. Muscles produce large amounts of heat during activity. The liver, brain, heart, and kidneys are also significant heat-producing organs at rest. Temperature regulation is therefore not only about staying warm in cold environments. It is also about losing excess heat generated internally. Sweating, skin blood flow, breathing, posture, clothing choices, and behavior all contribute. Humans are unusual among mammals in their capacity for sustained sweating over large skin surfaces, which help make long-distance endurance activity possible in hot environments. The cooling system of the body is one of the hidden foundations of human mobility. The lymph node swelling during an infection are not simply inflamed lumps. They are sites of intense immune activity. Immune cells enter the node carrying fragments of pathogens, and lymphocytes scan those fragments, searching for a match. When the right lymphocyte recognizes its target, it begins dividing rapidly, producing a local expansion of cells specialized for that threat. The swelling is partly the physical consequence of immune activation and cell proliferation. A tender lymph node is therefore evidence of organization, not chaos. It is a biological meeting place where information from infected tissue is converted into a targeted immune response. The body's sense of thirst is regulated by osmoreceptors in the brain that detect the concentration of solutes in the blood, and by volume sensors that detect changes in blood pressure and fluid volume. When the blood becomes too concentrated or blood volume falls, the brain generates thirst, and the pituitary releases antidiuretic hormone, which signals the kidneys to retain water. Thirst is not simply dryness in the mouth. It is a centrally regulated drive based on the chemical state of the internal fluid environment. The body must keep that environment within narrow limits because every cell depends on the right balance of water and salts. The acid-base balance of the blood is regulated mainly by the lungs and kidneys. Carbon dioxide produced by metabolism dissolves in blood and forms carbonic acid. By breathing faster or slower, the lungs can remove more or less carbon dioxide, shifting blood pH within minutes. The kidneys regulate acid-base balance more slowly by excreting hydrogen ions and reclaiming bicarbonate. This two-organ partnership keeps blood pH around 7.35 to 7.45. Outside that range, proteins and enzymes begin to malfunction. Breathing is therefore not only oxygen intake, it is also acid regulation. Every breath participates in chemical stability. The human body uses calcium for far more than bones. Calcium ions are essential for muscle contraction, neurotransmitter release, blood clotting, hormone secretion, and intracellular signaling. Because calcium is so important, its concentration in the blood is tightly regulated by parathyroid hormone, vitamin D, calcitonin, bone storage, kidney handling, and gut absorption. If blood calcium falls, the body can release calcium from bone to preserve nerve and muscle function. This hierarchy reveals something important. The body prioritizes immediate physiological stability over structural reserves when necessary. Bone is strong, but it is also a bank the body can draw from to keep the chemistry of life working. The human body contains many feedback loops that maintain homeostasis, but homeostasis does not mean nothing changes. It means controlled change around a viable range. Blood pressure rises during exercise. Heart rate increases during stress. Body temperature fluctuates across the day. Hormones pulse. Blood sugar rises after meals and falls between them. The goal is not stillness. The goal is adaptive regulation. The body is stable the way a skilled dancer is stable, through constant adjustment, not through rigidity. Life is controlled instability, continuously corrected. The body's relationship with sleep is deeply physical. Muscles lose tone. Body temperature drops slightly. Heart rate and blood pressure generally decrease. Breathing becomes slower and more regular in non-REM sleep. Hormones shift. The immune system changes its pattern of activity. The brain cycles through stages that support memory, emotional processing, and metabolic clearance. Sleep is not a shutdown. It is a different mode of operation, one in which many repair and regulation processes become more prominent. The conscious mind disappears for a while, but the body continues its work with a different emphasis. Dreaming occurs in the body, not only in the brain. During REM sleep, most voluntary muscles are inhibited, preventing dream actions from being acted out. Breathing can become irregular. Heart rate may fluctuate. Genital blood flow increases in both males and females, independent of dream content. The eyes move beneath closed lids. The brain generates vivid experience while the body is held in a strange state, internally active, externally still. Sleep reveals that consciousness, movement, and bodily arousal can be separated in ways waking life usually hides. The body's repair after exercise is not merely the healing of damage. Exercise creates signals. Mechanical stress, metabolic demand, calcium flux, and local inflammation all inform muscle, bone, blood vessels, mitochondria, and connective tissue that adaptation is needed. Muscles increase protein synthesis. Mitochondrial capacity can expand. Capillaries can become denser. Bones can remodel. Tendons can strengthen slowly. Fitness is therefore not stored in one place. It is distributed across tissues, each responding to the repeated message that the environment requires more capacity. Training is conversation with the body through stress and recovery. The human body also has limits built into its protective systems. Fatigue, breathlessness, pain, and discomfort are signals that help regulate behavior before catastrophic failure occurs. During intense exercise, the brain integrates signals from muscles, temperature, blood chemistry, motivation, and expectation to regulate effort. The feeling of exhaustion is not just the muscles running out of fuel. It is the nervous system's judgment that continuing at the same intensity may threaten homeostasis. This does not mean fatigue is imaginary. It means it is intelligent. It is a protective experience generated from bodily information. The body uses sleepiness as a signal, but sleep pressure has a biochemical component. One important molecule is adenosine, which accumulates in the brain during waking as ATP is used. As adenosine builds, it contributes to the feeling of sleep pressure. Caffeine works partly by blocking adenosine receptors, reducing the subjective feeling of sleepiness without removing the underlying need for sleep. During sleep, adenosine levels decline. This is one reason wakefulness cannot be extended indefinitely without cost. The chemistry of being awake leaves traces, and sleep is part of how the brain clears them. The human body is constantly balancing repair and defense with the risk of overreaction. The immune system must attack pathogens but tolerate food, harmless microbes, and the body's own tissues. The clotting system must seal wounds but keep blood fluid. The nervous system must detect pain but not amplify ordinary sensation into suffering. The stress system must mobilize energy but not remain activated forever. Many diseases arise not because a system is absent, but because a useful system is too weak, too strong, mistimed, or misdirected. Health is less like having powerful systems and more like having well-regulated ones. The body's internal environment is sometimes called the milieu intérieur, a term associated with the 19th century physiologist Claude Bernard. He recognized that cells live not directly in the outside world, but in the internal fluid environment the body maintains around them. Temperature, pH, oxygen, glucose, salt, and countless other variables must remain within workable limits. The outside world can change dramatically, but the cells need continuity. Much of physiology is the art of protecting that internal sea. The skin, lungs, gut, kidneys, liver, blood, and nervous system all participate in keeping the world inside the body habitable for its smallest living units. The human body is also a record of evolutionary history. The spine bears traces of a body plan that evolved for four-legged movement and was later adapted to upright walking. The recurrent laryngeal nerve takes a long looping path from the brainstem down into the chest and back up to the larynx because of how nerves and blood vessels developed in our fish-like ancestors. Goosebumps raise body hair in a reflex that made more sense when our ancestors had thicker fur. The appendix, once dismissed as useless, is now understood to have immune and microbiome-related roles, but it still reflects a digestive history different from modern diets. The body is not perfectly designed from scratch. It is inherited, modified, repurposed, and full of historical compromises. The upright human posture is biomechanically expensive and extraordinary. Balancing on two legs requires constant small adjustments from the feet, ankles, knees, hips, spine, eyes, vestibular system, and brain. The arches of the feet act as springs and shock absorbers. The pelvis supports abdominal organs and provides attachment for muscles that stabilize walking. The spine curves in several regions to distribute load and maintain balance over the hips. Walking looks simple because it is automatic, but it is controlled falling, a repeated movement in which the body tips forward and catches itself step after step. Every ordinary walk is a precisely coordinated negotiation with gravity. The human foot contains 26 bones, 33 joints, and more than 100 muscles, tendons, and ligaments. Its arches store elastic energy, adapt to uneven ground, and distribute body weight. The toes help with balance and propulsion. Sensory receptors in the soles provide continuous information about pressure and surface texture. Shoes can protect the foot, but the foot itself is an active sensory and mechanical structure. When you stand in the dark, small changes in pressure under the feet help the nervous system keep you upright. The ground is being read through the skin. The body has a remarkable capacity for redundancy. There are 2 kidneys, 2 lungs, paired adrenal glands, paired ovaries or testes in typical development, 2 cerebral hemispheres, and many overlapping biochemical pathways. Redundancy does not mean these parts are unnecessary. It means survival is improved when function can continue despite injury or variation. 1 kidney can often sustain life. A person can lose part of a liver and regenerate. Blood vessels can form collateral routes around blockages. Neural networks can sometimes reorganize after damage. The body is fragile in some ways, but resilient in others, built with backup capacity in systems where backup was evolutionarily valuable. The sensory world of the body is filtered. The brain receives far more information than consciousness can hold. Signals from the skin, joints, organs, eyes, ears, nose, tongue, blood vessels, and immune system flow continuously. Most are processed without awareness. You do not feel your blood pressure moment by moment. You do not feel your kidneys filtering. You do not feel most immune activity. Consciousness receives a curated summary. Pain when attention is needed, thirst when fluid balance requires action, hunger when energy is needed, fatigue when recovery is due. Much of the body remains silent precisely because it is functioning well. The internal organs also send sensory information to the brain through interoception, the sense of the body's internal state. Signals from the heart, lungs, gut, bladder, blood vessels, and immune system contribute to feelings such as fullness, nausea, breathlessness, heartbeat awareness, warmth, chill, and vague bodily unease. Interoception also shapes emotion. Anxiety can be amplified by sensing a racing heart. Calm can be supported by slow breathing and a relaxed gut. The boundary between bodily state and emotional state is not sharp. Feelings are partly interpretations of internal physiology. The vagus nerve is one of the major communication highways between the body and brain. It carries signals from the brain to the heart, lungs, and digestive organs, and carries a large amount of sensory information from the organs back to the brain. It helps regulate heart rate, digestion, inflammation, and aspects of stress recovery. Slow breathing, especially with longer exhalations, can increase vagal influence on the heart, contributing to a calmer physiological state. This is one reason breathing practices can affect emotion. They are not merely psychological tricks. They engage real autonomic pathways connecting lungs, heart, brainstem, and body. The body changes after a meal in a coordinated way. Blood flow to the digestive tract increases. The pancreas releases insulin. The stomach stretches and signals fullness. The small intestine releases hormones that slow stomach emptying, stimulate digestive secretions, and communicate with the brain. The liver begins processing absorbed nutrients. The immune system samples food-associated molecules and microbial material without overreacting. The nervous system shifts somewhat toward parasympathetic activity, supporting digestion. The familiar sleepiness after a large meal is not just laziness. It reflects a whole body shift toward processing, storage, and internal work. The body's water content changes across age and tissue type. Infants have a higher percentage of body water than adults. Muscle contains much more water than fat. Blood plasma, lymph, interstitial fluid, intracellular fluid, and cerebrospinal fluid are all part of the body's water compartments, but they are not interchangeable pools. Each has a specific composition maintained by membranes, pumps, channels, and pressure gradients. Water moves by osmosis, following solute concentrations, and the body regulates this movement through salts, proteins, hormones, and kidney function. Hydration is therefore not simply the amount of water present. It is the distribution of water in the right compartments. The body uses pressure in many ways. Blood pressure moves blood through vessels. Osmotic pressure helps regulate fluid movement between compartments. Air pressure gradients move breath in and out of the lungs. Pressure in the eye maintains its shape. Pressure in joints changes with movement and load. Pressure receptors in the skin detect touch, while pressure receptors in blood vessels detect circulation status. The body is constantly measuring forces. It is chemistry, but it is also physics, pressure, tension, flow, diffusion, elasticity, leverage, friction, and vibration. The human body contains many molecular clocks and counters. Cells can sense nutrient availability through pathways such as insulin signaling and mTOR. They can sense energy stress through AMPK. They can detect oxygen levels through hypoxia-inducible factors. They can detect damage through P53 and related systems. These pathways do not operate in isolation. They integrate signals about food, oxygen, stress, growth, and repair, helping the cell decide whether to divide, rest, repair, produce proteins, conserve energy, or die. At the cellular scale, life is decision-making without consciousness. The body's relationship with oxygen is paradoxical. Oxygen is essential for efficient energy production, yet oxygen chemistry can also produce reactive molecules that damage proteins, lipids, and DNA. Cells manage this through antioxidant systems, including enzymes such as superoxide dismutase, catalase, and glutathione peroxidase. These systems do not eliminate oxidation entirely, because reactive oxygen species also serve signaling functions at controlled levels. The goal is balance. Too much oxidative stress damages tissue. Too little reactive signaling may also impair normal cellular communication. Oxygen is both fuel partner and chemical risk. The body is constantly rebuilding proteins. Proteins fold into specific three-dimensional shapes that allow them to function as enzymes, receptors, transporters, structural fibers, antibodies, and motors. Misfolded proteins can be useless or harmful, so cells have quality control systems, including chaperone proteins that help other proteins fold properly and proteasomes that degrade damaged proteins. Autophagy, another recycling process, allows cells to break down larger structures, including damaged organelles, and reuse their components. Cellular health depends on renewal at the molecular scale. Even stable tissues are full of turnover. The motor proteins inside cells are astonishingly small machines. Kinesin and dynein walk along microtubules, carrying cargo such as vesicles, organelles, and molecular complexes. Myosin interacts with actin to produce muscle contraction, but myosin-like motors also function in many non-muscle cells. These proteins convert chemical energy from ATP into mechanical movement. Inside a neuron, materials must be transported along axons that can be more than a meter long. Without intracellular transport, distant parts of the cell would starve or fail. Even stillness at the level of the whole body contains motion at the level of the cell. The human body produces and responds to electrical fields in multiple forms. The heart's electrical activity can be recorded as an electrocardiogram. The brain's synchronized electrical rhythms can be recorded as an EEG. Muscles produce electrical signals measurable by electromyography. These signals are not metaphors. They are physical consequences of ion movement across membranes, coordinated across tissues. Medicine can read these patterns to infer rhythm, seizure activity, sleep stage, muscle activation, and nerve conduction. The body's electricity is quiet, low voltage, and biological, but it is real. The body's final common pathway for voluntary movement is the motor neuron. Upper motor neurons in the brain send commands down to lower motor neurons in the spinal cord and brainstem, which directly activates skeletal muscle fibers. Each lower motor neuron and the muscle fibers it controls form a motor unit. Small motor units, such as those controlling eye movements or fine finger movements, contain relatively few muscle fibers, allowing precision. Large motor units, such as those in powerful leg muscles, contain many fibers, allowing force. The nervous system grades movement by recruiting different numbers and types of motor units. A gentle touch and a heavy lift are variations in recruitment, timing, and force. The eyes are connected to the brain through the optic nerves, but part of each visual field crosses at the optic chiasm. Information from the left side of the visual world is processed primarily in the right hemisphere, and information from the right side is processed primarily in the left hemisphere. The brain then reconstructs a unified visual scene. This crossing arrangement reflects the deep organization of vertebrate nervous systems. Vision feels seamless, but it is assembled from two eyes, two retinas, crossed pathways, parallel processing streams, motion detectors, color processing, edge detection, memory, expectation, and attention. Seeing is not passive. It is active interpretation. The nose warms, humidifies, and filters air before it reaches the lungs. Turbinates inside the nasal cavity create turbulence, increasing contact between air and the mucosal surface. Blood vessels warm the air. Mucus traps particles. Cilia move mucus toward the throat, where it can be swallowed and processed by stomach acid. Nasal breathing also supports nitric oxide delivery from the nasal passages into the lungs, where it can influence blood vessel tone and gas exchange. The nose is therefore not just an opening for air or a smell detector. It is part of respiratory conditioning, immune defense, and airflow regulation. The body's connective tissue forms a continuous structural network. Fascia surrounds muscles, groups of muscles, organs, nerves, and blood vessels. It transmits mechanical forces, allows gliding between tissues, and contains sensory nerve endings that contribute to body awareness and pain. For a long time, fascia was treated in anatomy as material to cut away in order to see the important structures beneath. More recent research has recognized it as a biologically active tissue with mechanical and sensory roles of its own. The body is not assembled like separate parts in a bag. It is bound, layered, and tensioned through living connective tissue. The immune system must tolerate food. Every day, the gut encounters foreign proteins from plants, animals, fungi, and microbes, any of which could theoretically trigger immune attack. Instead, the gut-associated immune system usually learns tolerance to harmless food antigens while remaining ready to respond to pathogens. This requires specialized immune cells, regulatory signals, mucus barriers, microbial interactions, and constant sampling of gut contents. Food allergies occur when tolerance fails and the immune system mounts a response against a harmless food protein. Ordinary digestion, therefore, requires immunological restraint. Eating is an act of controlled exposure to the outside world. The body also has a chemical sense of oxygen and carbon dioxide. Specialized chemoreceptors in the brainstem and in the carotid bodies near the major arteries of the neck monitor blood gases and pH. Carbon dioxide is especially important for the drive to breathe under ordinary conditions. When carbon dioxide rises, acidity increases and breathing is stimulated. Oxygen becomes a stronger driver when levels fall significantly. This is why holding the breath becomes uncomfortable, mainly because carbon dioxide accumulates, not simply because oxygen has already run out. The urge to breathe is a chemical alarm generated by the body's monitoring of its internal atmosphere. The human body is warm because it is alive, but warmth must be distributed. Blood carries heat from active organs and muscles to the skin, where it can be released. When cold, blood vessels in the skin constrict, preserving heat in the core. When warm, they dilate, allowing heat to escape. This is why skin color can change with temperature. The body prioritizes core temperature because enzymes, brain function, and organ systems depend on it. Fingers and toes may become cold not because the body has forgotten them, but because it is protecting the center. The body's relationship with gravity begins before birth and continues until death. Bones, muscles, blood vessels, balance systems, and posture all develop and function under constant gravitational load. In spaceflight, without normal loading, muscles atrophy, bones lose mineral density, fluids shift toward the head, and balance systems must recalibrate. Astronaut physiology reveals that many features of the body we take for granted are responses to living in one gravity. Standing, walking, blood pressure regulation, bone strength, and even the shape of the spine are all Earth-adapted solutions. The human body contains traces of ancient viral infections in its genome. Endogenous retroviruses are sequences derived from viruses that infected the germline cells of distant ancestors and became inherited as part of the genome. Some of these sequences are inactive fragments. Others have been repurposed by evolution. One famous example involves syncytin proteins, derived from retroviral envelope genes, which play roles in placental development by helping cells fuse. A viral mechanism once used for infection became part of mammalian reproduction. The body is not only descended from animals. It carries molecular fossils of ancient encounters with microbes, some harmful, some eventually transformed into tools. The human body is also shaped by cooperation with other organisms. The mitochondria inside cells descend from bacteria. The microbiome helps digest food and train immunity. Viral sequences in the genome have been repurposed. Even oxygen-rich metabolism depends on photosynthetic organisms that transformed Earth's atmosphere long before humans existed. The body is therefore not an isolated achievement. It is a meeting point of evolutionary histories: bacterial, viral, animal, ecological, planetary. To understand the body fully, science must look beyond the skin. The final minutes before sleep are a physiological transition. Light fades. The circadian system permits melatonin release if conditions are appropriate. Body temperature begins to drift downward. The sympathetic system may quiet if the environment feels safe. Thoughts may loosen. Sensory attention narrows. Muscles settle. The body does not fall asleep all at once. Systems shift gradually into a new state. The brain will pass through lighter sleep, deeper sleep, cycles of memory processing, bodily regulation, and perhaps dreams. Sleep is not the opposite of bodily activity. It is the body's nightly change of priorities. The body lying here tonight is doing all of these things simultaneously. The heart is beating, the lungs expanding, the kidneys filtering, the liver processing, the bone marrow producing, the immune system surveying, the gut communicating, the skin sensing the room, the brain shifting slowly towards sleep. None of this requires instruction from the conscious mind. The body simply continues its ancient work, maintaining the conditions that allow the experience of being alive to continue. There is something profoundly calming in that. The body is not perfect, and it is not invulnerable. but it is extraordinarily competent. It has been built by deep time to regulate, repair, defend, adapt, and begin again. It turns food into movement, breath into chemistry, sensation into experience, injury into repair, darkness into sleep, and sleep into restoration. You do not need to manage any of it tonight. You do not need to remind the heart to beat, or the lungs to breathe, or the cells to continue their quiet work. The body has been doing this for you since before you had words for it. It will keep doing it as awareness softens and the edges of thought begin to loosen. And for now, that is enough. Sleep well. Good night.